Our next speaker is Professor Stephen Long from Memorial Sloan Kettering Cancer Center. So having trained with Rod McKinnon, a pioneer in ion channel structure, Dr. Long's research integrates structural and electrophysiological approaches to gain insights into the regulation and function of ion channels. Dr. Long has pioneered the structural biology of several ion channels, including ORI, which lies at the heart of calcium signaling in immune cells, as well as bestrophin and MCU, which is the mitochondrial calcium uniporter. Dr. Long has kindly agreed to introduce the audience to the most salient aspects of ion channel structure and function. Thank you, Dr. Long.
Thank you very much. I was remarking to my wife that I feel like I can only disappoint in a way because there is a lot here, and I'm sure that I haven't referenced everyone I should, so please accept my apologies in advance. But it's really great to be here today and to try and address ion channel structure and function. So some of the things that I'd like to cover, just an introduction to the property of some of the ion channels, just trying to continue on what Dr. Jen was presenting. There'll be a little bit of overlap, but not too much. And then look at sort of a case study in potassium channels, because the architecture of a potassium channel is related to lots of different types of cation channels. But that's not to say that all cation channels do look like potassium channels. And so perhaps another case there would be looking at the crack channel, or I, which our lab works on, and then present an example of an anion channel structure. And so the idea will be to try and understand how structure informs understanding of these channels. And some of the big challenges in this field of structural biology, well, one is obtaining a structure, obtaining that first structure of an ion channel that you don't know what it looks like yet. And then subsequently trying to understand, well, what does it look like under different conditions? One of the, I think, really interesting things about ion channels to me is that they do things. They move, they have parts, they not only let ions go through, but they have things like opening and closing, and they have domains that do different things. So trying to obtain structures that represent these different so-called gating states. And then even, I think, in a way more difficult is putting those into functional context. What are we looking at? And I think that's an important thing to think about really anytime that you're looking at a structure of an ion channel or of any protein. What is the structural biology? What does that thing you're looking at represent in terms of physiological state? And I also like to point out that I think in a way we're all structural biologists. We process the world around us in 3 dimensions, or at least we sort of naturally understand things. As children, you try and understand how a car works, perhaps, or a bike. And as structural biologists, we do essentially do that on the atomic level by taking a look at proteins at the atomic level to try and understand just how they function. And so what I'm showing here in the gray mesh is the density, the cryo-EM density. So this is density that was obtained using an electron microscope. And into that is built an atomic model. And so I'll just repeat that. So you can see that there are regions that, for example, form helices. This is a helix right here. And then you can sort of like building a puzzle, put the atomic model, since we know proteins are made up of a linear chain of polypeptide of amino acids. We know the sequence of those amino acids. We can essentially build the puzzle and make an atomic level model of the protein we're interested in, and then try and understand how it works, and then try and make experiments, design experiments to test some of our ideas about how it works. And so these are some of the ion channels that are present in the immune system, and we've talked about a number of these already. This is from a review that Stefan wrote, and you can see that there are a lot of ion channels in T cells. And I'll be touching on a few of them, and I sort of marked with a box here ones that are I call them KV-like. So these are, in a way, their transmembrane region is related, or it's in some way similar to a potassium channel, in that it's essentially 4-fold symmetric, has an ion conduction pore right down the middle, and has a similar domain type of structure. But there are definitely not only nuances, major differences between the different types of channels. But since so many channels are like a potassium channel in their transmembrane architecture, I thought we'd start with potassium channels. And then I'll also talk some about the channel that we've worked on a fair amount, the crack channel ORI. And then at the end, also touch on a chloride channel, which has a very different architecture. So just as a review, ions are stable in water, but not in oil. And we all know this from salad dressing. So if there's an oil phase and an aqueous phase, the ions go to the aqueous phase. And that's because ions are not stable in a hydrophobic environment. And that's really shown, I like to show this because it's just so dramatic. So this represents a synthetic bilayer here, so synthetic lipid membrane. And oxygen, nitrogen, carbon dioxide, these essentially go through pretty much unimpeded. They're easily diffusible through the membrane. And some of the permeability coefficients are shown here on the right. And whereas ions like sodium, chloride, calcium, potassium really are essentially excluded. And so you can see the difference between a sodium ion about 10 to the minus 12th for permeability coefficient compared to water is a factor of 10 to the 10. Which means that on their own, only about one ion would pass a membrane every 10 hours or so. And so it's extremely slow. And so you need a way, cells need a way to allow ions to flow. And of course, this is a very useful property because now we can have action potentials and we can have voltages across membranes and things like this, and then control them using ion channels and ion pumps. And so ion channels have, very simplistically, ion channels tend to have three general properties. And so this depicts, a concept of an ion channel in a membrane. It contains an ion conduction pore, which in this case is allowing blue ions to flow through. And the ion channels tend to conduct ions extremely rapidly. So on the order of 10 to the fourth, all the way up to 10 to the eighth ions per second. And the 10 to the eighth is really quite amazing because that's essentially the rate at which ions could reach let's say they're flowing in, if it's a sodium channel, they can reach the outside entry of the pore that's essentially the rate at which they can diffuse. So as soon as they are able to diffuse to the pore, they're able to continue diffusing right through the membrane, right through the pore. And so you can just comparing that to the one ion every 10 hours or so to one ion or 10 to the 8 ions per second, you can see just how good an ion channel is at catalyzing the flow of ions across membranes. But not all ion channels are extremely fast, and I think the crack channel is one it's important that it's slow, and we'll get to that later in the presentation, and it's more on the order of 10 to the fourth ions per second. Ion channels also tend to be selective, and certainly some are very selective, some are less selective, but they tend to be very selective. And so this kind of raises a major conundrum. How do you achieve such a high throughput rate of 10 to the 8th ions per second and yet be selective? When an antibody binds to its antigen, it does so by grabbing onto it and holding onto it very tightly and doesn't really let it go very much. And of course, in this case, the channel needs to let go of the ions about 10 to the 8th per second. So an enzyme, a very good enzyme might catalyze a chemical reaction at about 10 to the 3 times per second. So this is 5 orders of magnitude faster than that kind of thing, and yet it's extremely selective. So that's been a conundrum. But I think we understand some general principles of how that works. And then there's also the property of gating, which is depicted here at the bottom, which is just a conceptual depiction in that there's a part of the channel that essentially opens and closes and either permits or prevents ions from flowing. And these gates can be in different locations, and you'll see some examples of that in the presentation. So like Dr. Jen was saying, you can measure the currents that are carried by the ions flowing through even a single ion channel. And so here, in this case, the upper level here, we're looking at a single ion channel, and the current that's carried by the ions as they flow through the channel. And so this is the open state, and this is a closed conformation, a closed state. And so this is just to show that these gating conformational changes that we're going to be looking at might underlie these kinds of transitions that we can observe, macroscopically, that you can observe using electrophysiological rig. So again, I'd like to start with these KV-like channels and then start looking at just what potassium channels look like. So in the 1980s, by Dr. Jen and others, several potassium channels were cloned, and it was recognized that they all contain this so-called pore domain. It's a very simple type of architecture of two helices called the outer helix and the inner helix, separated by a short intervening helix that doesn't cross the membrane. So it has two transmembrane helices. But if you look at different types of ion channels, ones, let's say, that are voltage dependent, they have an additional bit here, which is the voltage sensor. Others might have a ligand binding domain, in this case attached to the C terminus. And so in a way, the channel genes are somewhat modular. You have a pore section and you have different parts that are responsible for governing whether or not, or biasing whether or not that pore is open or closed. And so we'll look first at just the structure of the pore domain and work that was done by Roderick McKinnon and eventually earned him the Nobel Prize several years later. This is the structure of the potassium channel KCSA. And you can see the approximate boundaries of a membrane here. The channel is comprised of 4 identical subunits. And like all of these types of channels, they either have fourfold or pseudo fourfold symmetry, depending upon whether or not, how many subunits are included in the gene. You can see here on the upper right, this is the structure of the channel viewed from the inside of the cell. But here, looking at it from the side, and we've stripped away two of the 4 subunits. So you can see that inner helix and the outer helix. Here's the pore helix. And then this section of the channel turns out to be the part that's responsible for selectivity. So we call that the selectivity filter. And at the time when this structure was determined, it wasn't quite clear what conformation it might represent. Is it an open conformation? Is it a closed conformation? And actually, if you go read the paper, it actually suggests that it might be an open conformation. And so it wasn't until a couple of years later when they observed this structure that they started to say, well, maybe this is open and this one is closed. And that, of course, is what we understand now, that this is closed and this is open. And so comparing these two conformations, which are actually of different channels. So again, it's very difficult. It has been very difficult historically to obtain structures that represent different gating conformations of the same channel. And in this case, both of these were crystallized proteins using X-ray crystallography. And KCSA happened to crystallize in a closed conformation, and MTHK happened to crystallize in an open conformation. Both channels are prokaryotic potassium channels. But you can see by comparing these two structures that, well, one, the region that I've mentioned is the selectivity filter essentially stays fixed between the two structures. But there's very large motions in the inner helices, and these inner helices get closer together to one another in the what we call the closed conformation, and further apart in the open conformation. And in the open conformation, there's now no impediment to ion conduction through the membrane, conduction through the pore. And we'll talk more in detail about how the ions get through the selectivity filter in a moment. But this sort of was, this was the first structure of a potassium channel. And it really gave us the first glimpse of what an ion channel would look like in 3 dimensions. And so it established some concepts that you have a selectivity filter that is essentially fixed, and you have a gate that is essentially mobile. And that turns out to be generally true for ion channels, but not exclusively true. Sometimes the selectivity filter is affected by whether or not the gate is open. And there's evidence that that's probably the case for the calcium channel Ori. So from these studies, there's these 2 conclusions that I point out. And the gate, it's these large motions. And so if you compare the structures in the upper right-hand corner, again, from the extracellular side, from the intracellular side, this is closed, and then this is open. So you can see there's a very big widening. And it turns out this gate, the one that closes, tends to be hydrophobic. And Stephen Tucker's lab has written a review on so-called hydrophobic gating in ion channels. The gate region that sort of seals shut the channel tends to be hydrophobic in nature. That's not exclusively true, but that tends to be the case. So now I'd like to focus on ion selectivity, just to try and point out some principles for how potassium channels are selective for potassium. And so focusing on this region of the channel, which contains ions in the selectivity filter, and also an ion here in the central cavity, which it turns out is surrounded by water molecules. And so hopefully you can see this movie. And this movie really gives me the chills every time I present it, because here is an ion essentially in water. So you have a potassium ion surrounded by 8 water molecules, and 4 of those are above and 4 of those are below. And so if we freeze it for just a moment here, the 4 molecules above are in a square configuration, and the 4 molecules below are in a square configuration, and those 2 squares are rotated with respect to one another. And the potassium ion is coordinated by these oxygens. And so now when we start to look at the region of the channel that conducts potassium, the selectivity filter, you can see that the potassium ion here is actually surrounded by 4 oxygens. And these oxygens actually come from the carbonyl oxygens of the backbone of the polypeptide. So that was actually a big surprise. But these carbonyl oxygens, they're 4 above, they're 4 below, and they're rotated as if to mimic this water structure. So as the ion moves through the selectivity filter, water gets removed from it, or most of the waters get removed from it. And the channel overcomes the energy barrier of dehydration essentially by substituting for those water molecules as the ion moves through. Now there are four densities. That's what this mesh represents. This represents electron density for the ions in the selectivity filter. And there are 4 of them. And it turns out that probably within a given selectivity filter, all 4 sites are not occupied simultaneously, but rather it's a little bit more than 2 of them. And they tend to be occupied in the 1, 3, or 2, 4 positions. And this ability to toggle between these sort of 2 configurations is thought to be really integral to how you can get selectivity and high conduction, the idea of having multiple ions in the pore. And there's some papers written down there at the bottom if you'd like more information on that. And there's been a lot of work done subsequently as well. So now I'd like to turn to taking this general architecture of a potassium channel and looking at what a voltage-gated potassium channel looks like, because all the ion channels that we mentioned in the slide a moment ago in the T cell that are related to potassium channels actually have a voltage sensor domain, or they have a voltage sensor like domain, something that sort of looks like a voltage sensor. And so we'll take a look at that voltage sensor domain, looking at the potassium channel KV 1.2. So again, voltage-dependent potassium channels have a pore region, which is composed of the S5 and S6 helices, or 2 transmembrane helices and an intervening pore helix. This will look familiar. And then it has this voltage sensor region. And the 4th helix of that contains positively charged amino acids that turn out to be the voltage, that they turn out to actually physically sense the voltage and actually move as the voltage changes. And it's these movements of the voltage sensor that govern whether or not the pore is open or closed. And so we were looking at how a pore might open and close. And in a voltage-gated channel, it's the voltage sensor that governs that process. Now, of course, the S4 helix is a really unusual thing. It's very unusual to have positively charged residues right in the middle of a transmembrane helix. And so there's interesting questions about how, for example, arginines are stabilized within the context of a membrane that I'm not really going to touch upon, but it's a very interesting question. So this just highlights conceptually the process. And so if we look at a channel in the closed conformation, which we call the resting conformation, even though it's actually higher energy than the open conformation. And that's because the resting conformation is at the resting voltage of the cell, which is about minus 70 millivolts. And so that minus 70 millivolts pulls down on the voltage sensors essentially and closes the ion conduction pore. And then when the voltage becomes less negative on the inside, the channel can open and become activated. And that's what's indicated on the right. And this is just a conceptual thing. This is not meant to indicate that the voltage sensors are sort of flipping upside down. So this is the structure, atomic structure of a voltage-dependent potassium channel, KV 1.2. And if you're not familiar with looking at these ribbon representations, each one of these ribbons represents a helix. And so there are six transmembrane helices per channel. We'll look at that in a moment. But you can see that the channel is a lot more intricate. It has a lot more pieces than that simple prokaryotic potassium channel. But a lot of its sort of core architecture of the pore is very similar, and we'll see that also in a minute. But this is the region of the channel that spans the membrane, and then you have these large cytosolic regions that I'm not really going to talk about much further. So looking at the channel from the top, one of the things that was a real big surprise about this structure was that the voltage sensors, which are, for example, here on the right, are actually quite separate from the pore. So they're actually separate domains from the pore, which was really not expected. And another thing is that the voltage sensor is actually next to sort of a neighboring subunit. So you have this, in a way, domain swapping type of arrangement where the blue subunit is actually next to the blue voltage sensor is next to the yellow pore domain, for example. But the pore shown in the middle contains a potassium ion here in the selectivity filter, and that's very similar in a way to the open structure that was visualized with MTHK. I'll just rotate this down, label the helices, and then rotate it down, looking at it from the side. So you can see behind this yellow voltage sensor is the pore architecture, the potassium channel, potassium ions in the selectivity filter, and then the voltage sensor, for example, here on the left. You can see the voltage sensor is next to one of the pore subunits. It's connected to the red pore subunit by this linker, the S4S5 linker helix. But really, it seems fairly daintily attached to the channel. And so a big question, of course, is, well, how do changes in membrane voltage cause the pore to open and close? Now, this was crystallized at 0 millivolts, because there's no membrane, and we didn't apply a voltage across the crystal. And so this was ostensibly be at 0 millivolts, which voltage you'd expect the channel to be open. And that's in fact what we see. The arginine residues are closer to the extracellular side, which in this case would be up, and the pore is opened as well. But how might how might the channel close? So this is looking at the pore. So we stripped away the voltage sensors, one of which would be here. The pore is open and conductive. So potassium ions in the cellular setting would typically move out, but potassium channels are passive devices, and if you apply appropriate voltages, they can move in either direction. And so the pore is open in this regard. The inner helices are wide enough to allow ion permeation. And we have a sense of what a closed pore would look like from the structure of closed pores like KCSA. So that's essentially what this would look like, where we've closed the blue inner helices and then maintained this interaction that we observed between the S4S5 linker helices and the S6 inner helices. And you can see that they're toggling between these two. So the S4S5 linker helices, these are particular to this voltage gated channel. It turns out that it's a very common feature among voltage gated channels. And we observed this interaction between these two helices that was proposed to potentially act like a lever as the voltage sensors get pulled toward the inside. When under resting conditions, that is again negative on the inside, you'd have a downward pull on those arginine residues in the S4 helix that would push down on the N-terminal end of the S4S5 linker helices and squeeze shut the ion conduction pore. And that seems to be, in fact, the case. Of course, it's very difficult to, I was mentioning how one of the big challenges is to obtain different conformations of ion channels. And I think especially for voltage-gated ones, because, technically you can't really apply a voltage very easily in any kind of structural biology setting. There are about 10 to the 14 ion channels in a crystal. And so I was calculating one day, I think what the voltage would somehow have to be, in around 30,000 volts or so across a crystal, which of course is not really achievable, even if we did have a membrane in there. And so there are other ways that one might think about trying to obtain a structure of that resting conformation. And this was a series of 2 papers done by different groups to try and achieve that for a voltage-gated sodium channel, which conceptually is, architecturally is actually very similar to a voltage-gated potassium channel. You have voltage sensors, you have a pore, you have arginine residues that sense the voltage and move. And in this case, what was done was actually to take voltage sensors from a mammalian channel, attach them to a prokaryotic voltage-gated sodium channel, and then in one case, they introduced a toxin, which actually stabilizes the voltage sensors in a down conformation or a resting conformation. And in another case, they introduced mutations into that prokaryotic channel that lock it in a closed conformation. But this allows us to get a visual on things that might be occurring. And so this is a movie between the two structures. So this is again the open structure, very similar to the potassium channel one that I showed you. And then this is a movie showing a transition to the resting structure, which is the closed one. And so you can see this downward motion of the S4 helix, and these blue spheres represent the arginine residues. And it's the pull on the arginine residues due to the electric field that sort of yanks down on the voltage sensor and that part of the voltage sensor, and then makes it move. And that causes sort of a clenching or a wrenching type of motion on the inner helices of the pore, allowing it to close. So, and that's just the reverse there. So things get even more complicated. Our channels have lots of bells and whistles, and in a way, the first one was like your simplest, maybe go-kart, and then we maybe looked at a entry-level car, and now this is sort of this is like the Bentley. This has got all the bells and whistles. This is the IP3 receptor. And there's been a few groups that have determined structures of this. It's really amazing. Just a few years ago, even thinking about trying to structurally look at the IP3 receptor seemed incredibly daunting because it's just such an enormous channel. This is the channel that's responsible for releasing calcium, one of the channels responsible for releasing calcium from the endoplasmic reticulum. So IP3 binding to this channel activates the channel, causing release of calcium from the ER. And the channel is enormous. It is a tetramer, as shown here. And this is a movie that I'll show you in a moment by Richard Heitzlab, who's right next door here to me at Sloan Kettering. And this is the EPO conformation of the channel. And then I'll start a movie and you'll see different conformations. But we're looking at the channel now from the inside of the cell, looking sort of down on the ER. So, and then at the middle here, you can probably recognize the fourfold symmetric pore. So this is the pore part, which is very similar in architecture to the pore that we've been discussing for a potassium channel. This, of course, is a calcium channel. It's not particularly selective for calcium, but it allows calcium to go through. And since there's a large gradient of calcium across the ER membrane, the calcium flows through in an appreciable way into the cytosol. But understanding how this channel works is really complicated. It's actually not in a way, it's much more complicated than a voltage-gated channel, because this channel is activated by IP3, and it's also activated by low levels of calcium, but it's actually inhibited by high levels of calcium. And so how do all these things come together? And a lot of the answers to those questions still remain to be determined. But this is just to show one of the conformational changes that can occur. And so this is the conformational change from calcium-free to calcium-bound. And so you can see there's this large rotation. I can't even describe it. It's what you see there to find calcium bound here. And this is the high calcium concentration. So this would probably be a calcium inhibitory site. And then you can also ask the question, well, what happens with IP3? And then there's this additional one. But so far, the fully open structure where you have a conductive ion conduction pathway through the channel, an open pore, has been elusive. And so that's one of the major questions is, even though we know a lot about the structure of the channel and we know now, where the calcium binding site is, which wasn't known before, we don't know really fully how this channel opens. And another thing that is really, I think, so powerful about electron microscopy. So that was electron microscopy, and the structure of this was enabled by electron microscopy. But electron microscopy, since you're looking at individual channel molecule, looking at individual proteins, you can then, if you have the right signal to noise, and perhaps if the molecule is big enough, like this IP3 receptor, it's kind of paradoxical, but bigger things are in a way easier to study with electron microscopy because you can recognize them more easily. But because of this, you can start to decipher nuances of the channel, and that's sort of shown in this movie, which is going from the apo structure to one in which IP3 is bound, and you'll see different things occur. There's actually different portions of the molecules that have 5 different classes, 6 different classes, I guess it is, where you can see that it's not a concerted motion. You have states which are fourfold symmetric in the apo and in this what's called the class 2, but along the way you can have these varied states where one of the 4 subunits is moving it's a neat thing. Of course, these are morphs. These are just theoretical extrapolations between fixed states that we observe. So these classes are fixed states or these are states that we observe, and then there's a morph that's made between those. So now I'd like to sort of having just barely covered potassium channels, turn to the ORI channel, the crack channel ORI, which is near and dear to us. And I think is a really interesting channel too, for several reasons. One, it's actually just so different than a potassium channel, not only in structure, but also in function. And so this channel is called the calcium release activated calcium channel, because calcium release from the ER, shown here, actually activates the calcium channel ORI in the plasma membrane. And unlike a lot of cellular calcium channels, ORI is extremely selective for calcium. It's one of the most selective calcium channels known. And another really interesting thing about it is that it's actually the crosstalk of these two membranes that opens the channel. So the ER membrane has a protein in it called STIM, and STIM has a calcium sensor domain that's located in the lumen of the ER. And when calcium is depleted from the ER, say due to the opening of the IP3 receptor, there's a conformational change that occurs in STIM that we still fully don't understand, that allows STIM to physically interact with ORI across the gap between the two membranes. And so the physical interaction between these two proteins residing in different membranes is what causes the channel to open. And that was really astounding to us, just that, how could that be? How could that occur? Now, this occurs at sites where the membranes are pre-existingly close together, so they're close enough for the STIM molecule to reach up and touch ORI. But that process is something that we're still investigating, and of course, lots of others are investigating too. And some of the labs are just mentioned at the bottom there. But across the plasma membrane, there's an enormous gradient of calcium, about 2 millimolar on the extracellular side and about 100 nanomolar on the intracellular side. And so there's large gradient of calcium, also in the context of about minus 70 millivolts resting voltage on the inside of the cell, creates an enormous driving force for calcium entry. And calcium is actually very different than its role physiologically is different than potassium, because potassium, as it flows, tends to change the voltage on the inside of the cell relative to the outside of the cell, whereas calcium is actually itself a physical signaling molecule. So calcium binds to other proteins like calcineurin and does things. And so very small changes in the calcium concentration, which are detected by these high affinity binding proteins, is the signal. And so calcium is really, in a way, the simplest signaling molecule that one might imagine. It's just an ion. And so this channel, in a way, because of this different physiology, the channel actually, and this enormous driving force for calcium across the plasma membrane, the channel, I think, would probably have to have a different type of architecture, because it's actually very slow. And I think it's intentional that it's slow, because if the channel were extremely good at conducting calcium would rush in, perhaps overwhelm the cell with calcium. And calcium is actually not even very soluble in the cytosol, because it would precipitate with the phosphate that's there. And so, you wouldn't want a very high concentration of calcium very quickly. And so the channel actually generates sustained calcium signals that can last for hours. And so you have a slow calcium influx. And so partly because of that, we thought the structure would be different, and it would perhaps reveal some barriers to ion permeation. And so this is the structure of the channel ORI that Xiaowei Ho and I determined several years ago. It turned out to be a hexamer, which was unexpected at the time. There's a single calcium shown here. We're looking at the channel from the extracellular side. It has actually a pretty simple architecture, with the M1 helices in blue lining the ion conduction pore right down the middle of the channel, and the other helices sort of arrayed in sort of concentric rings around the M1 helices. And we'll rotate it down, looking at it from the side. You can see that the channel really doesn't have much that extends into the extracellular solution, but extends pretty far into the intracellular cytosolic solution. And that's in line with the idea that it interacts with STIM, which is reaching up from the ER membrane. Again, the ion conduction pore is right down the middle, and it's a very simple type of architecture, essentially lined by these M1 helices. And I'll just zoom in on those M1 helices, looking at the ion conduction pore, and then compare that to to the type of channel we were just looking at before, a voltage-gated potassium channel. And so you can see that the pores are actually very different in their chemical and dimensional environments. In Ora, you have a very long, slender pore, and the pore chemical environment is dictated by the amino acid side chains that emanate from the M1 helices and line the pore. Whereas in the voltage-gated potassium channel or other potassium channels, you have the selectivity filter that's familiar, and then a very wide open aqueous cavity through which ions can permeate. And so looking in more detail at these amino acids reveals sort of these sections of the pore. There's only a single glutamate residue, only a single negatively charged residue on the pore. Of course, there are 6 of them due to the 6-fold symmetric nature of the channel. We're looking at 2 of the 6 subunits here from the side. And then below that, there's this very unusual hydrophobic region, and the hydrophobic region spans about 2/3 or about half or so of the membrane. And it's interesting because this hydrophobic region, you'd imagine, would it turns, if you do a membrane, a secondary structure prediction and looking for transmembrane helices, the M1 helices come up as transmembrane helices, and they do so because of these hydrophobic residues. It actually has some hydrophilic residues which point out and are actually sort of encased by the other helices of the channel. But the hydrophobic ones are the ones that actually line the ion conduction pore, which is kind of interesting to us. But I agreed very well with Morali's data of cysteine scanning mutagenesis. But it's an unusual feature for an ion channel to have these hydrophobic section right at the middle of the membrane. And then below that, maybe even more surprisingly, were 3 basic residues from each of the 6 subunits, which creates an environment with 18 basic residues. And of course, these point in to the center of a channel that's supposed to conduct a positive ion, calcium. So you have this, the idea that you have these enormous potentially energy barriers, one, a hydrophobic energy barrier, just because you have ion dehydration, and that might occur, and there's an energy cost associated with that. And then perhaps an electrostatic barrier as it reaches the basic region of the pore. Now, of course, this is a closed conformation of the channel because STIM is not present. And so this is sort of a concept. Another thing that was a really big surprise that I can't quite tell if the field doesn't believe us or just no one has really looked into it further. We observe that there's very strong density for iron, Fe, in the middle of the basic region. There's very good data for that from our structure. And also, this is of the Drosophila channel. We also have the human channel with iron in the pore as well. And this iron is probably bound as iron hexachloride, which forms a complex, almost like a complex anion, and plugs the pore, and I think would need to be released for the channel to open. And so, one question is, well, how does the channel open? And to try and get at that, one of the things that we've been doing is to look at mutations that have been identified by others that cause the channel to be constitutively active, that is active without having to have STIM present. And one of those that we focused on in the Drosophila channel is H206A. And that one is nice because it's not on the M1 helix, it's on the M2 helix. And so this mutation doesn't line the ion conduction pore. Some of the mutations that cause it to be constitutively active line the ion conduction pore. And those are problematic, sort of for obvious reasons that if you change the ion conduction pore, you might be changing the properties of the channel in a really dramatic way. But there's good evidence, there's decent evidence that, well, this H206A constitutively active mutant generates an IV curve, a current voltage relationship that's similar to the STIM activated channel, suggesting that it's similar to the STIM activated channel. Of course, we want to observe how STIM actually activates this in a more physiological way. But in any case, this allowed us to visualize an open conformation of the pore. And again, just to try and to get an idea of how this thing might open. And this was also done using electron microscopy. In this case, we made antibodies that helped us to determine the structure at 3.3 angstroms resolution, which was sufficient to observe amino acid side chains and their conformation. And so from that, if we look at the hydrophobic section of the pore, this is just to give another example of what gating can look like in an ion channel. And so the first structure was the closed, and then this is moving to the open. And so you can see that hydrophobic section of the pore, and in fact, the entire pore widens. It widens more so on the cytosolic side than it does on the extracellular side. The selectivity filter doesn't widen very much, but there does seem to be a slight change in selectivity filter due to positioning of the calcium ion within it. But the hydrophobic section probably functions essentially as the gate of the channel, and that's not just from our work, but from others as well. And this is an indication of how that might work. And a thing that's, I think, really interesting about it is that as it goes from the closed to the H2O6A open conformation, you don't see rotations of helices. In fact, the subunits essentially move independent of one another, essentially away from the center of the pore to allow the hydrophobic region to get wider. And you can see this phenylalanine residue, for example, it's just undergoing a small rotation of its side chain, but we don't see a twisting rotation or a twisting motion of the helix along its axis. So now I'd like to just briefly touch on an anion channel, just to highlight the fact that they have a very different architecture. And there's something about anion channels that each family seems to be really quite different than the next. And so, so far I've shown ion channels that have ion conduction pores down the middle, but that's not necessarily the case. The ion channels, the CLC family of ion channels, they're actually dimers, but they have two independent pores that are within each of the sort of halves of the full molecule. And in other channels, like the TMEM family that we were just discussing, those ion conduction pathways are actually on the edges, sort of at the interface between the membrane and the protein. So it's not even going through a continuous pore that's surrounded by amino acids. It's actually going through sort of this crevice. And that's a really interesting thing. So anion channels, like Dr. Jen was mentioning, chloride is the predominant physiological anion that would be permeable through these channels. And so since there's really only one anion, I think there's different architectures that allow it to go through. The other anions in the cells are things like, are bigger, like RNA molecules or mRNAs and things like that. Of course, you have ATP. So anyway, so I'd like to show the chloride channel, the LRRC8A family chloride channel. And this is a structure that was by Ryman Dutzler, but there's been a few other structures that are very similar to it. And this ion channel is interesting for several reasons, but one, it's interesting because it's involved in volume regulation. It's actually activated by low ionic strength, and that seems to occur through changes in this part of the protein down here, more toward on the intracellular side. So this again is the approximate dimensions of the membrane. This would be the extracellular side. This is the intracellular side. But it's not yet clear, and I bring this up, partly just to highlight one of the things I was mentioning at the beginning, which is that it's difficult sometimes to associate a particular structure with a particular functional state. Maybe you observe a particular functional state, and then correlating that to the structure is difficult, especially until you're able to get different structures that say, okay, that one is much bigger, much wider pore than this one. And okay, I can now understand that the channel undergoes inactivation. And so now I say, okay, my structure originally was inactivated. Now let's try and get an activated structure now that I understand, the properties of inactivation. So I'll just show this movie. This is a six-fold symmetric, well, it's actually three-fold symmetric, but it has 6 subunits. And the ion conduction pore is right down the center. Then there are these large leucine-rich repeat domains on the inside. And if we just zoom in on a region of the channel that is thought to be the selectivity filter out here, you can see it has some basic amino acids that seem to underlie its selectivity for chloride. But like a lot of chloride channels, this channel is not very selective for chloride. So potassium channel is quite selective, and ORI is extremely selective for their particular ions. Chloride channels tend to be not that selective. So for example, they might permeate nitrate or carbonate or thiocyanate. And these are useful things for the electrophysiologists studying them, because you can look at different ions and their properties. But some of them have physiological functions and other ones maybe not, in terms of the particular ions that are moving through. Okay, so there's a lot to be discovered. There's a lot to be discovered from a structural biology perspective. There's a lot to be discovered, certainly from a functional perspective, about how all these channels in immune cells work. And I'm excited about that. This is a COVID-style picture of my laboratory where people have been photoshopped in, but we are happy to be open and fully in person and doing science. There are positions in the laboratory available. And I never thought I'd live in New York, but it's been a great place to work. And I'd also like to and live. And I'd also just like to point out some resources that I think are quite valuable. The first one is this website that categorizes all membrane proteins for which the three-dimensional structure has been determined, and that's really nicely it's very nicely organized. So that's a great resource for looking at ion channels. And then this program, maybe I'll start with the program at the bottom called PyMol, which is quite user-friendly. You can download this, academic versions of this, and run them on your computer. And this allows you to visualize atomic models and make really nice figures. And especially if there's a structure that has been determined for a related ion channel that you're interested in, you could pull it up in PyMol, spin it around, and maybe you'll come up with new ideas about how it works that haven't been talked about in the paper. And also, I'll bring up UCSF Chimera. This is a program that's useful for visualizing electron or cryo-EM density, the density that you get from an electron microscopy analysis. And that's really important, I think, because there are a lot of structures coming out now, cryo-EM structures, and not all cryo-EM structures or any structures are sort of equal in terms of their quality. And so I think it's important to look at, what does a 4 angstrom structure look like? What does a 3 angstrom structure look like? What does a 5 angstrom structure look like? And especially if you're interested in a region of the molecule, it's a good idea to just pull it up and look at it and say, okay, well, actually the density is not that great for that region of the molecule. So maybe it could be modeled incorrectly, or it could be flexible. And those things might inform your thinking on how the channel works. So with that, I'd be happy to take any questions.
Thank you, Steve. That was an excellent talk. We have a bunch of questions. One question that had come up was the involvement of lipids in ion channel gating. So obviously, a lot of these ion channels make contacts with a lot of lipids. And as you know, some of these structures don't incorporate some of these lipids. So how do you think about addressing those issues? And this has actually become quite an important factor for trip channels.
Yeah, absolutely. So, yeah, lipids like a soluble protein requires water. Ion channels and membrane proteins require lipids. Well, first, I'd maybe try and address a structural biology perspective. We have very good detergent molecules. So the detergents we use are not Triton X. So the detergents are generally very gentle. And so there are very, very few examples where the detergent causes dramatic. There are a few sort of notable examples, but there are very few examples, statistically speaking, where the detergent causes a notable sort of distortion of the membrane protein. And from my own lab, we have now determined structures in a number of cases in both detergent and lipidic environments, like nanodiscs, where the structures are indistinguishable from one another. But certainly lipids play a major role in ion channels, and certainly in regulating certain ion channels of the voltage-gated potassium channel-like family. There's a lipid that I could go back, to this slide. There are lipids that bind between S4 and the S4S5 linker, for example, and different members of these families that modulate the function. In the K2P family of ion channels, these are very well known to be modulated by lipids. Even lipids might be involved in the ion conduction pathway of certain ion channels, because they've been observed in the structures, essentially openings in the transmembrane region, which are directly exposing the ion conduction pore to the lipid membrane. And so you can imagine a direct interaction between lipid molecules. And the sort of the dawn of cryo-EM is allowing us to try and allowing us to control that a little bit better using electron microscopy, because we can use what's called a lipid nanodisc, which is a protein scaffold that surrounds the transmembrane region of a protein, into which you can incorporate lipid molecules, and you can choose the lipid molecules you want to incorporate, and then obtain a structure that's in a a semi-membrane-like environment. And I think it is important to remember that it is still a semi-membrane-like environment. It has probably a lipid bilayer right around the protein, but it's actually probably not that different than a detergent environment in the sense you have acyl chains and you have hydrophilic stuff. And the acyl chains are closer to the membrane protein, and the hydrophilic stuff is further away. In the case of the nanodisc, it's the scaffold protein in the head groups of the lipids. But if you want to observe a particular lipid, that can be useful. And also in certain, at high enough resolution, you can often resolve lipids. And so in the structure of KV 1.2 that we determined, we actually resolved lipids in the X-ray structure, and those sort of generally resemble a bilayer. But yeah, I think that's in a way a big next frontier to structural biology, and also to understanding the function of ion channels in general, just controlling the lipid environment, studying that probably in a reconstituted system where you can more easily control the lipid environment from an electrophysiological perspective, that is, so studying, how do lipids affect the function and how is that related to the channel? Yep.
There was a question on IP3 receptor, and I think the question deals with the IP3 receptor hinge and how much of that is electrostatic versus just a binding of IP3 in terms of some kind of steric change.
Okay, yeah, this is one of the dangers of presenting someone else's data. So I don't know the answer to that question particularly well. Maybe the others do that are on the call, but
no worries. Yeah, we have a bunch of questions on actually ORI channels, and those might be interesting to look at. So if in the case of ORI, do the mutations in ORI2, which increase Socky, do they alter the conductance of the channel?
Yeah, that's I don't know the answer to that question.
Okay.
Sorry, maybe I'm sure Rich Lewis does, so he can chime in.
I think we will ask him that. Was ORI mutant structure crystallized in presence of calcium, the open ORI?
Yes.
And how much calcium?
Yes, crystallized. And so we have both a crystal structure at about 7 angstroms resolution that's published in eLife, I guess 2018. And then or maybe 2019, and then we have this is a cryo-EM structure, and the crystal structure. One of the sort of limitations of cryo-EM is recognizing ions is a little bit more difficult than it is in X-ray crystallography, because X-ray crystallography we have the trick of using so-called anomalous diffraction of certain elements like barium, like gadolinium, like iron, and even calcium have these signals that allow you to pinpoint the location or to identify particular ions. In cryo-EM, we can observe their density. And so, yeah, all these structures were determined in millimolar concentrations of calcium. And we see calcium in the selectivity filter, basically in this position. Actually, in the open structure, it's a little bit lower. And that was related to my comment about the selectivity filter changing a little bit. The ion moves sort of more into the middle of these glutamates. But I would like to hasten to point out that although we see essentially one ball of density, I suspect there are probably 2 calcium ions in a permeant type of situation, which are probably the second one is occupied transiently. And so even though we only see one ball of density, there's good, I suspect there are probably 2 binding sites.
Mm-hmm. There is a question wondering if there is progress on co-crystallizing ORI with CAD fragments of STIM or STIM. Yeah,
so we've been working on that for many years now. We're working on it, not too much progress.
Mm-hmm. In terms with the LRRC8, the structure you showed there, it's an anion channel, but it's also considered a large pore channel, right? In the sense that it allows the permeation of cGAMP, and that's become very prominently, and it's very important for the innate immunity. So these, in the case of large pore channels, are these going through in a hydrated manner, these anions, you think? Are they partially hydrated, or what's going on there?
Well, I mean, I think our terminology of hydrated versus dehydrated is not so good, because I think oftentimes what people, at least the way it sounds, is that hydrated means, I guess, it's surrounded by water, and dehydrated means it's not. But even in the case of a potassium channel, the potassiums are not fully dehydrated. They probably have a leading and a trailing water as they move through the pore. And so I think that, if we take the case of an ion channel that we know better, which is bestrophin, which is a calcium activated chloride channel, there's a narrow region of the channel that we know is responsible for different anion permeabilities, anion selectivities, and that the anions go through at least partially dehydrated because the conduction sequence, the permeability sequence correlates to what's called the lyotropic permeability sequence, which essentially it goes with the dehydration energy. So that's an indication that the ions go through partially dehydrated. So I suspect that, yeah, definitely dehydration is probably occurring to some extent in every ion channel. And regions that are flanked in particular by hydrophobic sections, narrow regions that are flanked by hydrophobic amino acids, are probably more prone to effects of dehydration than ones that are hydrophilic.
Yeah. Great. Thank you so much, Dr. Long.